Thank you so much for having me. This is an amazing opportunity and an amazing meeting to be able to attend. So I'm going to talk about food-based foundations for the gut microbiome. I am primarily a gut person, so the things I say here apply mostly there. I have no disclosures to make. We're going to talk mostly about factors that are shaping your microbial community, some different ways to measure the microbiome, and long and short-term diet effects. So my first question is, what makes us human? Given what we talked about yesterday, you're probably thinking it's our genes, or our cells, excuse me. So if we start at the cellular level and we think about where we get most of the cells in our body, there are about 30 trillion human cells. There are about 39 trillion microbial cells. So we're only about 40% human cells. If we go on to our genes, about 20 to 30,000 human genes in our genome. This is compared to between 2 and 20 million microbial genes that are carried in our guts and around our bodies. So 2 orders of magnitude more microbial genes than human genes. Additionally, the level of diversity that we find between our human genes and our microbial genes is huge. So you share 99% of your human genes with every other person in this room, but you might only share 40% of your microbial lineage with the person sitting next to you. And that 40% of the microbial lineage misses additional characteristics of those genes that we're still working to understand. The microbiome has been linked to a huge number of diseases. I've picked a subset here. This list keeps expanding and expanding and expanding. And so it's becoming this both global problem. The other thing to think about is that it's a global system. Your microbiome is an ecosystem, making you this vast organism, this collection, this personal planet, if you will. And so as we're trying to map this and figure out what's going where, there's a series of new tools that let us do this. So most people, when they think of bacteria, probably think about old school, like agar cultures, where you're going into the lab, you're swabbing a plate, you're growing stuff up, and you're characterizing it. What percentage of the bacteria in the world do you think you can capture with this technique? 10%. Anybody? 50? 5? 1 to 2? So based on recent estimates, you're at about 1%. And you're looking at organisms in isolation, since the media will only grow sort of a single thing in a single place. In the body, it gets up to 30 to 40%, depending on who's doing it and what their techniques are. And so we're really only looking at a small fraction of the bacteria using these methods. Luckily, thanks to the sequencing revolution, we've been able to move forward from that. So when you talk about most typical microbiome experiments, you're looking at a 16S rRNA experiment. We're going into the bacteria and we're picking a specific gene. And in this case, it's the 16S rRNA gene. And this acts a little bit like a fingerprint for each bacterial species. So we can take these and we can actually build a universal tree of life based on that 16S rRNA gene. If we want to include mammals, we can throw in the 18S rRNA gene and we can see how related we are to dinosaurs or the E. coli that lives inside of you. And this combination of being able to sequence 16S rRNA and build a phylogenetic tree really revolutionizes our ability to understand the bacterial organisms that live inside of us. And so to be able to do this, you go out, you collect a sample. Hopefully you collect some clinical data along with it to give yourself context. And then you can take the samples and you can run about 600 samples on a MySeq run using barcodes. Process the samples, we use robots. and run them on a MySeq, and then analyze them using some standardized pipelines that do things like line up your sequences, build your tree, and make visualizations. And so this is sort of the overall view of what you're getting out in your microbiome experiment. When we do these experiments, we ask some big ecological questions. So what organisms are present in a microbial community? How similar or different are they? How do they compare to other communities around them? And how do the organisms interact? Right now, the first three questions are questions that we can answer relatively easily. And the fourth question is something that I think represents the next, hopefully, several decades of work from a career perspective, but maybe not from an overall perspective. And so I want to talk today about sort of the second and third points in this, because I think these are really important techniques to understand as you come at microbiome experiments and microbiome data. And so I think a lot of people come at this sort of from a foundation with Koch's postulates, where you're thinking, I want to find a single organism, the master organism that causes disease. Who is it? What are they doing? Can I supplement to change it, or can I remove it somehow? And I want to encourage you to think about this from a more holistic perspective in terms of a microbial community. So we're going to deal with a model community consisting of 3 organisms. These could also be bacteria, but I happen to like these 3 a little bit better. So we have these communities that consist of some mixture of the organisms. If we think about them in a context, we may have 4 communities that consist of 4 different mixes of organisms. And if we think about it simply from a diversity, how many organisms are present in each community, it should be relatively easy to identify which community here is the most diverse and which community here is the least. And so if we go through and we do this, you can count up the number of organisms and identify that the purple community, third from the left, is the most diverse. The community to the far left is the least. And then we have a tie in the 2 communities, more in the other 2 communities. And this is a measure of alpha diversity, which is a question of how similar or different the organisms are in a community. So the metric we just looked at is something really simple, counting up the species, which would be observed species or observed OTUs. We might want to bring a different perspective to this, though, because maybe we have some additional hypotheses about what matters in communities. And so, okay. So there you can see that we're counting up the organisms, and we're getting this simple measure. We can consider something like relative abundance of different organisms using a quantitative measure. This is going to be something, as you read the literature, like Shannon or Simpson diversity, that accounts for richness, the number of species. And so here, if you look at this, you can see that the community on the left has better distribution of organisms compared to the community on the right, where the community on the right is dominated by 1 species compared to the community on the left. This effect of dominating the community makes it less diverse. And this is a characteristic you commonly see in disease. So like in Clostridium difficile infections, you see an overgrowth of C. diff compared to other members of the community. And that's part of what drives the loss of diversity in that disease. A second way to look at diversity is to consider evolutionary history. So go back to that phylogenetic tree that we're able to build and compare the organisms. And so we can take into account this phylogeny and look at the metrics, and we can actually build up based on those metrics and count. And so as we consider diversity metrics, there are 4 primary types. They're quantitative, they're qualitative, they're taxonomic, and they're phylogenetic. And as you're reading the literature, there are sort of different ones that you'll come across. Faith's PD is a phylogenetic unweighted. Shannon is a taxonomic weighted, and those are probably the two most common you'll see. The second type of one of the major caveats as you're talking about alpha diversity, though, is that you can have communities that have completely different organisms, share absolutely nothing, and they will have the exact same diversity measure. So this is not a comparison of the organisms across community, simply the organismal counts. On the flip side, alpha diversity is a really nice variable to be dealing with because you're not making that big comparison. The second type of comparison you may want to make, though, is a between community comparison. How do the organisms from one community line up with another? And so if we go back to our model communities, we can count up what's there. And we can just say what fraction of each community is similar or different. And so here you can see that the two communities on the left share a larger fraction of their organisms than the two communities on the right, based on weighting them by abundance. And this is the formula for a metric that you'll run into a lot in microbiome literature, Bray-Curtis. You, again, might want to consider the phylogeny, the shared evolutionary history, where you'll count up the shared branch length and compare that to unique branch lengths on the tree. This is the unifrac metric. With unifrac, you can also weight it by abundance. Full disclosure, I'm from Rob Knight's lab. We use unifrac a lot from the perspective that this evolutionary history really does matter. As we do this, we make those pairwise comparisons between each organisms, and we can actually start to build up a matrix that compares each sample by each sample. And eventually we fill this in. And we use this, and we can take the data and sort of twist it into three-dimensional shapes to make these plots, principal components plots, sometimes also seen as cloud plots. So here I've taken the data, I've turned it into a plot. We do this completely agnostic of clinical information. And then I can paint on clinical information for the samples. So I can say that some of them in red are from the US and some of them are from Venezuela. And you can see that there's this big split between the two sets of samples based on the country of origin. There is, of course, one caveat, because everything has caveats, which is that as you introduce new samples to your data set, you can shift this visualization. And so the plots that you show are dependent on the samples you're seeing, and inclusion or exclusion of samples can drive observed clustering. So now that we have some tools, let's look at early studies of the microbiome and where we're going with these. So one of the first major explorations was the Human Microbiome Project, which was $100 million on about 250 healthy adults from 2 sites in the US. Most of these healthy adults were medical or graduate students at the sampling sites. And so most are in their 20s and 30s. Most are relatively healthy or completely healthy by clinical exam. We don't know anything about their diet. We don't know much else about them. When this was done, they got a series of sequences. And here is a snapshot of the first sequencing file. And I'm sure looking at this, you guys can tell that it's an oral sample. So we take the data, we run it through the pipeline, and we're able to do principal coordinates analysis. And here's the plot we get, no clinical information painted onto it. They went through the clinical information, they started painting things on, and they found that there's this major effect of body site. And so you can see that oral samples cluster together, skin samples cluster a little bit with urogenital samples, and then fecal samples exist sort of in their own landscape. This was great in 2012, but it's not really informative today. So there started being more questions. Is this the whole microbial world? Is everything summed up in those 250 fecal samples? And so a second study about the same time looked at fecal samples from 3 populations: the US, Venezuela, and Malawi. And there are some major cultural and dietary differences between these 3 countries. The US has sort of more of a westernized diet, and the populations that were looked at in Venezuela and Malawi have more traditional diets. So when you take that data and you do the principal coordinates analysis on it, you can see that there's a separation, that samples from the US cluster separately from samples from Venezuela and Malawi, which cluster together. Where do you think the HMP data falls within this sphere? Do you think it's the span of the US data, or do you think it's a subset? So if we take the HMP, it's only this tiny, tiny portion of the full data set, suggesting that there's a lot more of the world to explore. So shortly after this was published, Rob and a collaborator launched the American Gut Project. It's a crowdsourced, crowd-funded citizen science project. We have more than $2 million, and we've sequenced more than, I think at this point we have 15,000 samples, more than 100,000, or more than 10,000 participants, excuse me, and we're still collecting data. If you're interested in the project, you can check out our website, americangut.org. From a research perspective, we're actually making all of the de-identified data public. And so if you want to get into the data, it's publicly available. We have 200 self-reported variables. We've also added a validated food frequency questionnaire. And so especially for people who do nutritional work, this is hopefully a resource that can be used if you're interested. We have a bunch of covariates. A lot of them are food-related. And when we take this data and we include the American gut, shown here in light blue, with the data set I showed you before, we've actually shifted the variance in the data and what's explaining the difference. So most of the difference in the data actually comes from variance within the American gut compared to variance with Western, non-Western. And you can see that this is, in fact, an effect of the American gut data rather than an effect of something like study. Since you have those Western global gut samples from the US buried in the American gut cloud, whereas there's separation with the non-Western red samples. And so essentially, with this project, we've started to capture the world of Western gut samples and the diversity there. And so we can build on this to start asking questions about things like diet. So we asked a really simple question. How many types of plants do you eat in a week? This was 2012. We were a little bit naive. We didn't ask about servings. We literally asked how many types of plants. If you eat something that is a mix of carrots and peas, that is 2 types of plants. So we took the data, we calculated alpha diversity, and we started looking at the diversity between the samples. So people who reported eating fewer types of plants had lower diversity. People who ate more types of plants had more diversity. We sat there and we went, okay, well, what if the people who just don't like to eat plants have some characteristic about them that would affect their diversity in some other way? Maybe it's all babies, so we threw out all the kids. Maybe it's people who have IBD, so we threw out all the people who have IBD. And the trend holds. We also got some criticism saying, well, this isn't a great measure of what people are actually doing. So we took our validated food frequency questionnaire, and we compared the number of fruits and vegetable servings that people reported compared to the number of types of plants they reported. And we see this strong relationship. So we think that this variable is a proxy for vegetable servings and maybe a proxy for additional information there. When we compare this to the way people reported their diet, we actually find that there's less of an effect for people who are, vegetarian compared to the number of types of plants they eat. It's been proposed that you have the breaditarians and then you have the people who are actually eating vegetables. And so we think the difference may be from something like that. We can estimate statistical power by resampling the data and look at the power. So here we have the probability that you're going to see a difference on the y-axis and the number of samples you would need to see that difference on the x-axis. And so the farther over to the left a curve is, the more likely you are to see a difference, the bigger that effect. And so if we take known large effects, like age and country, we see that we're likely to see that difference. It's pretty easy. We don't need a ton of data. If we take something like our plant variety, it's a little bit shifted, but it's still a lot further to the left than diet. We need fewer samples to see it. It's a bigger effect on phylogenetic diversity. This is a cross-sectional study, but it lets us ask questions about things like community structure. So there was this great paper that came out last month that compared guts to coral reefs. And they did this amazing work with coral reefs. And we're able to come up with this hypothesis that all healthy communities are healthy in the same way, whereas unhealthy communities are unhealthy in their own way. In a very literary reference, it's the Anna Karenina principle. And so we think that high diversity communities are healthy and resilient, like a high diversity coral reef. But as you start to get insults, like antibiotics or like a poor diet, you start to build up these impacts. And just like a coral reef can bleach, you start to bleach your gut, damage your diversity, and lose some of that stability. So from this, you can think about it, a diet rich in plants, whether or not you're including animal protein, is promoting microbial diversity. And this microbial diversity may be promoting community stability. One of the second major questions, I think it's everybody's question, is okay, can I change my microbiome? Is somehow this community that I've got my fate, or do I have an opportunity? And so there have been a couple short-term diet studies. These tend to be controlled feeding experiments. So they collect dietary information about a long-term diet, and then they get people in a controlled environment, feed them some food, maybe some different diets, and see what it does to their microbiome. And so there's an early paper where they said, okay, how does this shape our community? Well, they did principal coordinates analysis. They plotted the data, and they went, each person clusters by themselves, and we can sort of start building clusters on diet based on long-term dietary patterns. So here you can see that each person is colored differently, their collection of time points, and this pattern sort of clusters together based on the large diet. So, okay, maybe we are stuck. But a second study said, okay, if I take that data and I say I'm comparing me to myself, you can start to look first at the dietary composition. So they did a high fiber diet, a super low fiber diet, with varying amounts of fat. And they took the microbiome data and they looked at alpha diversity using a weighted taxonomic metric. And there's not a big change in alpha diversity on the top. But when they look at beta diversity, they find that the animal-based diet led to a shift in the microbial communities. And when they tested it, it was a significant shift in the microbial community, suggesting that there is a dietary influence. Our lab did a quasi-controlled feeding experiment where we recruited a group of different people and they were eating at home, but they were all on a standardized diet. I'm showing the data here from a popular juice cleanse. So if we color the data by individual, you can see that each person makes their own cloud. And then we can color it by time point. We can see that there's a difference before, during, and after. And actually, if we do a trace, you can see that each individual goes through a change in their microbiome, that they start out in the orange region, they move into the red, and then the blue moves back as they go off the diet. This suggests that communities go back to their previous state, and that diet is something you need to keep up if you want to see a change in the microbiome. I think there are a lot of people who are a little bit disappointed by this finding, who wanted to be able to, go on a week-long crash diet and suddenly have a major shift. And so your long-term microbiome, your microbial self, is really difficult to change over a short-term intervention. But you can alter, you can alter it short-term, but you need to do something persistent if you want to see a long-term change. Right now, the long-term studies are starting and in progress. Hopefully, we'll see data from these soon. The other major thing, though, is that we've done some of these in mice. So Justin Sonnenberg's lab at Stanford did this amazing experiment where they took mice and they either fed them a high-fiber diet or a low-fiber diet. And then they took the pups. And in the pups where the mother was fed a high-fiber diet, they split them. And they either fed them a high-fiber diet or a low-fiber diet. And in some cases, they transferred some of the low-fiber diet pups back to a high-fiber diet. And they saw this reduced recovery based on the maternal diet. And so low-fiber diet pups from moms who had been on a low-fiber diet lost the ability to recover diversity. There was a generational compounded effect from poor diet and poor food. And so this is something that needs to be considered as we're looking at current exposures and contemporary impacts on microbial communities and what we can do to recover. There are a bunch of people who have assisted with this work. I want to thank Rob and his lab. And thank you.